Yes,
Yes,
sir.
sir.
Okay, madam, recording. Okay, madam, record చేస్తున్నారా?
చేస్తున్నాను సార్.
Okay. Okay, good morning, my dear friends. Welcome to today's session in the faculty exchange program. Today, our topic is the another most important phytohormone, that is abscisic acid. So yesterday we have discussed about the cytokinins, and the day before yesterday, gibberellins and auxins also. Auxin, gibberellins, cytokines we have already discussed. Today we are going to discuss about the abscisic acid. Abscisic acid, ABA. In abscisic acid, we are going to discuss about the what is the history of this abscisic acid, what is the chemical composition of the abscisic acid, and where it is occurrence, where is the occurrence or occurred in which part of the plant, and distribution from the one part to another part of the plant, even though biosynthesis of the phytohormone, that is abscisic acid, and transport of the abscisic acid, and signaling pathway, signaling transduction pathway, and what are the roles of the abscisic acid in the different plants. Okay, today is our topic, that is abscisic acid, and history about abscisic acid, the beautiful hormone. This is a very interesting hormone. Regarding of the discovery of this hormone in 1940s, scientists are isolated the substance from the uh, sycamore leaves, called dormins. Okay, sycamore leaves called as the dormins. Hamburg is a scientist in 1940. These are the, along with this Hamburg et al., isolated the chemical substance from the sycamore leaves, named as dormins. And 60s, in the early 1960s, Eagle and Wearing et al., Philip Wearing et al., confirmed what Hamburg has isolated or separated from the substance in the leaves, sycamore leaves. The dormins, the application of this dormin to bud would induce dormancy. So bud dormancy, that is induced bud dormancy. So the dormin is able to or capable to induce the bud dormancy, identified by Philip Wearing et al. In the early 1960s. And 1963, Adicott et al. Is a scientist. Adicott et al. Scientist. The group of scientists, the Adicott and his followers, discovered a substance stimulated abscissin of cotton fruit. Unripened or immature cotton fruit. Cotton fruit we can call it as cotton bolls, cotton bolls. So immature cotton bolls, they are separated. Adicott et al., they are separated the chemical substance from that fruit, and that chemical substance is able to stimulate abscissin in the cotton fruit. He named this substance as abscissin 2. In 1964, it became evident that the three groups had discovered the same plant hormone, that is abscissin 1, abscissin 2, and abscissin 3. Whose work? 1963, Adicott et al.'s work again evidently proved that in 1964, in three groups, abscissin 1, 2, 3. And later on, the name was changed, that is abscisic acid. Abscissin is name has changed from abscissin to abscisic acid, ABA. Okay. Then, what is the chemical composition of this abscisic acid? The chemical composition of the abscisic acid is this abscisic acid is 15 carbon sesquiterpene compound. Yesterday we are discussed about the abscisic acid and gibberellic acid. We know that gibberellic acid the chemical structure, we know the 20 carbon compound or 19 carbon compound, we are already Ga3 and Ga3, gibberellic acid, 20 carbon compound, you know. At the same time here, the abscisic acid is the 15 carbon component, that is the 15 carbon component, that is sesquiterpene compound, sesquiterpene compound. This is composed of this chemical composition we are observed. This is composed of 3 isoprene residues. 3 isoprene residues. Then cyclohexane ring with keto. Cyclohexane ring with keto, 1 hydroxyl group. 1 hydroxyl group. 1 hydroxyl group, a side chain with a terminal. Carboxyl group. It is included even the carboxyl group. This is abscisic acid, the structure of the abscisic acid. And what are the formulas or synonyms or formulas of the abscisic acid are different names we are observing here. So ABA, number 1. The abscisic acid also ABA. Abscisic acid also earlier dormin, and earlier abscissine 2. Abscissine 2, dormin, ABA. So these are the synonyms for the abscisic acid, ABA or dormin or abscissine 2. These among these 3 are also nothing but abscisic acid. So these are the synonyms of the abscisic acid. And the chemical formula or molecular formula of the abscisic acid is C15H20O4. C15H20O4. This is the chemical or molecular formula of the abscisic acid. And the molecular weight of the abscisic acid is 264.32 grams of the molecular weight of abscisic acid. And this abscisic acid in external appearance, the external appearance of this abscisic acid is white crystallized in nature, white crystals. The purity of this abscisic acid, 98% purity we are observed. Then the melting point of this abscisic acid is 183 degrees to 180 degrees Celsius. So more than 100 degrees Celsius, you know, nearby 200 degrees Celsius is the melting point of this abscisic acid. And loss on drying more than 0.5%. Okay, more than 0.5%. And the chemical name of this is a chemical formula, molecular formula, and these are the synonyms of abscisic acid, ABA, dormin, and abscissine 2. Along with this, chemical name is abscisic acid, that is 2,6,4-trans, 5 1-hydroxy 2,6,6 trimethyl 4-oxo 2-cyclohexane 3-methyl 2,4-pentadenic acid. So this is the chemical name of the abscisic acid, you know, abscisic acid. Then the orientation of the carboxylic group at carbon 2 determines the cis and trans isomers of ABA. See, this is the cis ABA. This first image shows about the chemical structure of this abscisic acid. This is a cis position, cis ABA. Naturally, this is very natural occurring. It is an active form of abscisic acid. Then this trans ABA. So this is a 2 trans ABA is 2 trans ABA. This is an inactive but interconvertible with active cis form. So this is the interconvertible. So this is a isomers among these between these two structures interconvertible, but the cis ABA is only active. It is naturally occurring. Naturally occurring. At the same time, the trans ABA structure is inactive mode. So this is the active and this is the inactive mode. Okay. Then cis is a biologically active for the reaction in the physiological functioning of the different plants. Even though trans abscisic acid biologically inactive, but both of they are interconvertible. Okay. Nearly all the naturally occurring ABA is the cis form, not in trans form. Only cis ABA form occurring naturally, all the abscisic acid. And occurrence and distribution of abscisic acid in the plants. ABA is a ubiquitous plant. So ubiquitous plant hormone in vascular plants. So this abscisic acid absorbed in the vascular plants. Vascular plants from cryptogam, vascular cryptogams like pteridophytoplants to gymno and angiospermic plants is produce this abscisic acid. It is a ubiquitous plant hormone. In bryophytes, it has been found in mosses, but not in liverworts. Okay, actually, these abscisic acid found only the vascular plants only. So vascular bundles are steel, the stellar consisting plants only produce this abscisic acid. But even though some of the bryophytes like moss plants, not liverworts and hornworts, only mosses can able to produce or synthesize a little bit of amount of the abscisic acid. Okay, so actually abscisic acid is only absorbed on the vascular plants. Along with this vascular plants, only moss plants of the bryophyta we are observed, or liverworts and hornworts not able to synthesize this abscisic acid. And even though some fungi, along with this bryophyta mosses, along with these mosses, some of the fungi synthesize ABA as a secondary metabolite. So abscisic acid is synthesized in some of the fungi as a secondary metabolite we are observed. And 15 carbon compound called lunaric acid. Lunaric acid, A15C, 15 carbon compound called lunaric acid, has been found in algae and liverworts. My dear friends, remember what we are already earlier discussed here. At this point, only abscisic acid produced or synthesized only in the moss plants only, not liverworts and hornworts. But here, the 15 carbon compound lunaric acid, it is a one type of abscisic acid, but it is found in algae and liverworts also. ABA is synthesized in all types of cells that contain chloroplasts or other plastids. It occurs predominantly in mature green leaves. So abscisic acid synthesized in the pots related to all cell organelles like chloroplast and other plastids like alluroplast and elioplast also. So chromoplast and leucoplasts also contain this abscisic acid. And mainly the predominantly occurred in mature green leaves. Mature green leaves we are observed this abscisic acid. Has been detected, this abscisic acid detected all major organs or living tissues from root caps to apical buds. So entire part of the plants is has responsible for the detecting of the abscisic acid. Even though phloem sap and xylem sap and in nectar, right? Nectar also we are observed abscisic acid. And what is the biosynthesis? How this is the synthesis, the mechanism of this biosynthesis of this abscisic acid? The initial stages occur in the plastids. Initial stages, this is the plastid, This is the plastid, chloroplast. The structure is chloroplast, my dear friends. This image visible? Biosynthesis slide. This image visible, amma? This image visible? Amma? Nobody can respond. This image visible. So this is the plastid,
Yes,
plastid. Okay, very good. This plastid structure we are observed, this is the plastid. Then where isopentyl diphosphate, that is the IPP. So IPP, isopentyl diphosphate, is converted to C40, xanthophyll or geoxanthin. So this is the geoxanthin. So this is a geoxanthin, this is C40, 40 carbon component, geoxanthin. Okay, then geoxanthin again further modified into. So this is a pyruvate and glyceraldehyde is going to modify into IPP, isopentyl diphosphate. That is the intermediate. Then this isopentyl diphosphate is going to convert it into C40 40 carbon component, that is geoxanthin. Okay, xanthophyll geoxanthin. The geoxanthin further modified into 9-cis neoxanthin. neoxanthin. So 9 neoxanthin, which is cleaved by the enzyme NCED. NCED, that is 9-cis epizycarotenoid dioxygenase to form the C15 inhibitors xanthozole. So xanthozole, whenever the stress-induced reaction, whenever the stress-induced reaction we are observed, this trans neoxanthin or vioxanthin C40 is going to convert it into trans neoxanthin. This 9-cis neoxanthin C40 40 carbon component is going to modified in the presence of the NCED enzyme. Then again the xanthozole. So xanthozole is synthesized. So this xanthozole again, then converted into, in the presence of SDR, then ABA is synthesized. ABA in the cytosol. So ABA we are observed in cytosol, my dear friends. Cytoplasm. Cytosol means cytoplasm. The remaining all intermediates, all reactions taken place are taken inside the plastid, inside the chloroplast. Inside the chloroplast, not only chloroplast, but also remaining chromoplast or even the leucoplast also. So these chemical reactions or biochemical reactions we are observed in the plastid, and then afterwards this in the presence of NCED, this cis neoxanthin is moves towards the cytoplasm, and in the cytoplasm the xanthozole is finally converted into abscisic acid. So this is about the biosynthesis of the abscisic acid. Then mutants have been isolated that cause defects in the conversion of abscisic acid, that is the aldehyde into ABA, ABA aldehyde into ABA. Then flaca and cytinins in Solanum lycopersicum, NARA2A in Hardia vulgare, and ABA3 and AA03 in Arabidopsis. So these are the chemical components related to abscisic acid. So this flaca and cytins. Okay, flaca and cytins related to abscisic acid we are observed in the Solanum lycopersicum, Solanaceae family, Solanum melangina and Lycopersicum esculentum. These are the plants contain the abscisic acid, ABA modified structures, that is the flaca and cytinins, and NARA2A in Hardia vulgare, barley seed, you know, Hardia vulgare. Then ABA3 and AA03 in Arabidopsis thaliana. These are the plant shows this by ABA. And ABA transport in plants. ABA transport. How it is transported in the plants. Then externally applied ABA. Whenever we are applied externally ABA, it is distributed in all directions. Whenever we are spraying on the plants, whenever we are externally the chemical compound of ABA sprayed on the plants, leaves the chemical compound of the abscisic acid distributed in all directions of the plants, not in polar region, not in the polar directions, but all directions. And even though cell-to-cell transport is very slow compared to internal structures, the movement, the transportation of this abscisic acid is very slow from cell to cell of the internal, from the intercellular spaces. Then ABA synthesized in root cap, transported to central vascular tissue. Actually, generally ABA is synthesized to which part of a root cap of the root caps. Okay, and transported to central vascular tissue of the even though root xylem and different parts of the plants. Then afterward, this ABA transported mostly in its free form. ABA transported into free form, that is the transported in a conjugated form as ABA, beta D glucosyl ester. This is a conjugated form. ABA is a conjugated form, that is the ABA beta D glucosyl ester is a conjugated form. And redistribution of ABA in the presence of external pH gradient. Then the pH gradient is also responsible for the whether the low level pH or high level pH. At low level pH, then protonated or undissociated form ABAH. Even though at high pH, this is dissociated from ABA. this undissociated with ABA, that is a low pH, and dissociated ABA, dissociated form of ABA, that is at a high pH concentration. Then ABA signal transduction pathway. Then ABA signal transduction pathway, abscisic acid elicits two responses. Two responses, my dear friends. Number one, rapid response. Number two, gradual response. Rapid response and gradual response. These are the two responses we are observed of the abscisic acid. The first one is the fast and slow responses use different receptors. Different receptors. There are multiple abscisic acid receptors which are soluble and membrane bound. Multiple ABA receptors which are soluble and membrane bound. And this ABA, three classes of ABA receptors. Number one, the plasma membrane localized G proteins, then plastid localized enzymes, and the third one is the cytosolic ligand binding proteins, that is the start domain superfamily. So these are the three classes of ABA receptors. Number one, plasma membrane localized, that is nothing but G proteins, and plastid localized enzymes, and the last one is the cytosolic ligand binding protein, that is the start domain superfamily. And the first one is the soluble start proteins. These are the soluble start proteins are the principal ABA receptors that function in stomatal closing and germination. Stomatal closing and opening is most important regarding of the all physiological functions of the plant soma, just because of the exchange of the gases. Even though exchange of the gases in the process of photosynthesis, in the respiration also, the stomatal closing and opening is most important physiological function. That physiological function is determined by abscisic acid. Okay, so stomatal closing and germination of the seeds also responsible of this ABA concentration. And the first experimental performed in Arabidopsis thaliana. So Arabidopsis thaliana is a plant for the best performance of the first experiment on ABA, abscisic acid. Then pyrabactin. Pyrabactin is a synthetic compound similar to abscisic acid. So genes conferred in sensitivity to pyrabactin cloned called PYR1, that is pyrabactin 1. In Arabidopsis, PYR and PYL are start domains proteins and ABA dependent inhibitors of one class proteins phosphate type 2 PP2C, that is the protein phosphate type 2. See, and AB1, ABI1 and ABI2 member of PP2C. So if in different experimental approaches, this is the PYR and ABA transport in the central signaling module. This is the central signaling module. This is PP2C and SNRK2. We are clearly observed. This is the cytosol. This is the cytoplasm, and this is the nucleus. In the signaling pathway, how cytosol is shows different forms, and even though nuclear region. And what are the development and physiological roles of the abscisic acid? Then developmental and physiological roles of abscisic acid. Number one, promotes stomatal closing. Very, very important, because of that. This is a stress hormone, drought stress hormone. Okay, so this is a very, very important stress hormone, that is abscisic acid. This it promotes stomatal closing, and even though induce bud dormancy and seed dormancy. Induced bud dormancy and seed dormancy. The first one is stomatal closing, and the next one is the bud dormancy and seed dormancy induced by abscisic acid. And third one is promote desiccation tolerance in the embryo. So desiccation tolerance is the most important regarding our development of embryo, and which is the next one, the inhibits precocious germination and vivipary. Precocious germination and vivipary in especially Rhizophora avicennia or of halophytic plants are and this is very, very important of the vivipary, and promotes root growth and inhibits shoot growth in stressed water-logging condition, even though which is responsible for leaf senescence. So these are the important physiological role of abscisic acid. Number first important physiological role, that is stomatal closing. The opening of stomata, opening of closing of stomata is a shows explained about number of theories and Lloyd theories and sugar theories and potassium ion pump theory. Okay, so these theories explained about how this stomata is closing and opening. We are going to discuss in our physiology, plant water relations, and this stress hormone, that is ABA, is responsible for the closing of stomata. ABA, plasma membrane receptor, ABA. See here. So this is the plasma membrane. So membrane is contained. This is the channel. This is the channel for the potassium ion and calcium ion. How moves from the guard cell plasma membrane. Here we are observed. This is the opening state of the stomata, and this is the closing state of the stomata. Closing state of the stomata and opening state of the stomata. This stomata is whenever the potassium ion enter into the potassium ion enter into the stomata is opening. But this potassium ion is moves from outside from inside to outside of this guard cell, then automatically the stomata is going to close. So this is the inside and this is the outside. So water content, water molecules and potassium ion is moves from inside to outside, then automatically the guard cell, from the guard cell the stomata is going to close. At the same time, calcium ion is enter into the inside the guard cell. Calcium ion enter into the inside the guard cell whenever abscisic acid is a precursor or induce the potassium ion enter into the guard cell. At the same time, the potassium calcium ion enter into the guard cell, potassium ion and water molecule moves out from inside to outside, then stomata is going to close down. Here, the mutants that lack the ability to produce abscisic acid exhibit the permanent wilting and are called wilty mutants because of their inability to close their stomata. The stomatal closing can also be caused by abscisic acid synthesized in the roots and exported to the shoot. So actually, abscisic acid is synthesized in the root cap, my dear friends. Remember, root cap is responsible for the synthesis of the abscisic acid. Then afterwards, this abscisic acid moves towards the transported to the shoot regions. And the next important physiological function of this abscisic acid is induced seed dormancy and bud dormancy. And growth of the seed suspended dormant seeds, the primary dormancy and secondary dormancy is observed by the abscisic acid, and also which is controlled by the ratio of abscisic acid to gibberellic acid. Okay, then embryo dormancy is due to the presence of inhibitors, especially abscisic acid, as well as the absence of growth promoters such as gibberellic acid. Whenever gibberellic acid is absent, or at the same time presence of the ABA is responsible for the dormancy of embryo or embryo dormancy. The loss of embryo dormancy is often associated with the sharp drop in the ratio of ABA to GA. So between this differentiation of the concentration of the ABA and GA, then automatically GA is increased, the embryo dormancy is increased, then GA concentration is decreased, automatically the embryo dormancy is increased. So this is the variation. Model for tobacco seed germination. So this is the tobacco seed, the germination whenever the abscisic acid and remaining gibberellic acid or other phytohormones are responsible. So this is the promote plus or inhibit, the minus shows about the inhibit and plus shows about the promoting of the this is the compound state of the abscisic acid, that is the class 1 beta 1,3 glucanase index. Glucanase is a abscisic acid, and GA is gibberellic acid, that is gibberellic acid, gibberellins, ABA abscisic acid, then PFR phytochrome. So among these three phytochrome and gibberellins and the abscisic acid concentration. This is the working model. How this tobacco seed is going to germinate. We are the observe. This is the osmotic regulation here. This is the light. Whenever the presence of the light, PFR, PR going to modify it into PFR. Then gibberellic acid is induced the germination of the seed. So this is the coat of the seeds, testa and tegmen. These are the testa and tegmen. These are the two seed coats of the seed. And testa, that the seed coat is ruptured, then endosperm is going to protrudes or produce the little amount of the endosperm in the outside from the seed. So this is the day 1 and day 2, how it is responsible. And even though the endosperm of the sheath, then abscisic acid and ethylene concentration is we are observed here. Then the endosperm rupture, then endosperm weakening in the presence of the beta-1,3 glucanase. See here, this is the beta-1,3 glucanase, and day 6 again. After day 6 and the control day 6, then automatical germination of the stage, germination stage 3, we are observed the control, that the day 3 and day 4. And this is day 6 and day 6, how the germination is varies, we are observed. And induced dormance of the bud. Whenever we are the storage, where we stored in the dry conditions or the cool conditions in the cold storage regions, or in the room temperature in the room, the potato ice we are observed, the potato buds or ice we are observed in this slide or in this image. Then whenever we are applied in the externally this abscisic acid, then we are stored a number of days or number of days for the not able to germinate of these buds. The dormant buds, shortened internodes and especially modified leaves. So that is the bud scales. Then most of the 3 buds are formed late summer and remain dormant throughout the winter. Whenever the levels of endogenous ABA is high, then herbaceous plants, for example potato, we are observed here. So this is a woody plant bud. This is a woody plant bud. How bud is a dormancy state, and these are the buds in the dormant state of the potato tuber. Okay. And which is the promote desiccation tolerance in the embryo. So desiccate tolerance of the embryo. So these are the embryos, these are all embryos. This is the embryo of the in important function of ABA developing seed desiccation tolerance. The levels of ABA in seed peak during embryogenesis. During the mid to late stage of seed development, the specific mRNAs accumulate in embryos. These mRNAs encode so-called late embryogenesis, abundant LEA. So late embryogenesis abundant, that is LEA proteins. Okay, these mRNA encode we can call LEA, late embryogenesis abundant proteins. This LEA, synthesis of many LEA proteins induced by ABA treatment. And another important physiological function of this ABA is abscisic acid. It inhibits precocious germination and vivipary. When immature embryo are removed from their seeds and placed in culture media, they germinate precociously. Then ABA added to the culture media, it inhibits precocious germination. So this is the most important point regarding of the germination of the even this result in combination with the fact that the level of endogenous ABA is high during mid to late seed development suggests that ABA is the natural constant that keeps developing embryos in their embryogenetic state. During seed development, embryos of monocot and dicot plants may fail to complete maturation and germinate while the immature seed still attached to the maternal tissue. This phenomena is referred to as vivipary or pre-harvest sprouting. We are observed this pre-harvest sprouting or vivipary we are observed in the halophytic plants like Rhizophora avicennia, especially Sundarbans area. Plants are the seeds are not able to germinate in this high salinity content. So that's why within the fruit the seeds are going to germinate or seedlings, or whenever removed from this fruit, then automatically at the time of germination. So this germinated seeds we are observed. This maize, several viviparous mutant have been selected, which are the embryo germinated directly on the cob. So this is the kernel or cob of the geomesh, while still attached to the plant. Actually, these seeds are already germinated within the cob of the geomesh. Then vivipare in the ABA deficient mutants can be partially prevented by treatment with exogenous ABA. And the next important physiological function, that is the shoot and root growth. So shoot and root growth. ABA has a different effects on the growth of root and shoot, and the effect are strongly depends on water status of the plant. So the experiment done on the maize, in the maize plant, two types of the seedlings were used. Number one, wild type seedlings with normal ABA levels and ABA deficient viviparous mutant. So these are the two varieties of the maize plant, and this suggests that endogenous ABA promotes. The first one is, so endogenous ABA promotes shoot growth in well-watered plants. Whenever the water content is available, plenty of the water available and abundant available, then the endogenous ABA acts as a signal to reduce shoot growth only under water stressed conditions. Whenever water is not able to availability of the seeds, then automatically endogenous ABA acts as a signal to reduce the shoot growth. So this is the difference between availability of the external water content and the leaf senescence. So this is the most important physiological function regarding of the seed dormancy or induced dormancy, bud dormancy. Even the leaf senescence is also important. So the deteriorative processes that naturally terminate their functional life are referred to as senescence. So this expression of senescence associated genes, that is SAGs. SAGs means senescence associated genes. Whenever this increase, the senescence is high. Then hydrolytic enzymes, that is a proteases, and ribonucleases and lipases. These are the enzymes are responsible for the leaf senescence. And ABA involved in leaf senescence, that is the senescence of leaves, but not the abscission of leaves. So this is the main difference between senescent leaves and abscission leaves. So this is about the physiological functions of abscisic acid. If you have any doubt, please ask me, I will clarify. Abscisic acid. If you have any doubt, please ask me, Amma. Abscisic acid. The biosynthesis of abscisic acid and the physiological function of the abscisic acid and transportation of the abscisic acid. If you have any doubt, please ask me, Amma.
Girls, if you any doubt, ma? Girls, sir, no doubts.
Okay, madam. Thank you very much. Shall I leave?
Okay, sir. We will meet on Saturday, sir.
Okay. Thank you, madam. Thank you very much.
Okay, girls. Thank you, girls.